Well, good morning, everyone, and welcome to this series of lectures entitled Chemistry, a Hand Outreach to Society, which brings together different topics related to chemistry, but also to chemical engineering and biochemistry. Today, we are attending the ninth talk of this semester, and we will have the pleasure of having with us Mercedes Diaz-Mendoza from the Department of Biochemistry and Molecular Biology, who will talk to us about the useful expression system to produce recombinant proteins, plant cells. The title of this talk is Plants as Biofactories, Production of Proteins of Biomedical or Pharmaceutical Interest. As I always comment when presenting these talks, we have chosen the online format instead of the face-to-face format because in the context of the pandemic, this type of webinars were almost the only alternative. Therefore, we took advantage of the platform provided by the General Foundation of the UCM, whom I would like to thank again for their help and support in organizing this activity. This format is more suitable at the university because more people can be connected and even watch the videos that are kept on the Foundation YouTube channel. I am also grateful to our funding sources, the Spanish Society of Biochemistry and Molecular Biology, the Royal Society of Chemistry, the Madrid sectorial of the Vice Rector of Research and Transfer for the financial support. The choice of the language for this activity responds to the fact that we write, discuss, and disseminate science in English. Our junior researchers joined our institution after spending a more or less prolonged period abroad. Organizing these webinars in English can even make it easier for them to communicate their scientific results. In this way, we can broaden our audience for foreign institutions, which in many cases are the ones that have hosted these researchers. The talks will always take place on Thursday at 12:30. And the last one will be on the 2nd of June. And each webinar will be announced at the beginning of the week, including the link of the registration form. Without further ado, I give the time now to the moderator of this talk, Carlos Pastor Vargas, professor of the Department of Biochemistry also. Professor Pastor will introduce the speaker and moderate the questions that you can write in the preguntas and respuestas box that you have at the bottom of your screen. Now is your turn, Professor Pastor.
Thank you very much, Maite. Well, it's a big pleasure for me to introduce you to Mercedes Diaz-Mendoza. Mercedes completed her PhD in 2006 in the Plastic Interaction Laboratory at CIB in Madrid. Her dissertation was awarded a doctorate extraordinary prize by the Polytechnic University of Madrid. As part of her PhD, she stayed in 2005 at the University of Newcastle in UK. After the completion of her PhD, she started a postdoctoral period from 2006 to 2007 at the Veterinary Department in the Polytechnic University of Madrid. In 2007, she got a postdoctoral position at the CRAC in Barcelona to 2008. After, she spent 5 years of postdoctoral experience in USA from 2008 to 2013. Mercedes started working at the University of California in USA as postdoctoral research, and after the 3 years, she researched the next academic level as assistant specialist. In 2013, she got a position in a Marie Curie research in the biotechnology department in the CBGP in Madrid, granted by the People Program for Contracting Experienced Researcher under the Incoming and the Reintegration Marie Curie Action to 2016. In 2016, she was awarded by the Torres Quevedo Mineco program for contracting experience research in collaboration with private companies. During the last years, Mercedes has combined her research activities with private company collaboration and teaching at different universities in Madrid, Polytechnic University, Francisco Vitoria, Alfonso X, and the Universidad Complutense de Madrid. She got an assistant professor in position in 2020 at University Complutense de Madrid at the biochemistry and molecular department. Our department. Her actually research line is focused on the field of protein production in plants system to be used for molecular and structural characterization as well as biomedical and pharmaceutical purpose. Okay, Mercedes, up to you. When you want.
Okay, thank you so much, Carlos. I'm going to share the screen. And let's go start. Well, first of all, I want to thank all the organization for giving me the opportunity to be here presenting my work today. And also I want to thank Maite and Carlos for their kind presentations. Let's go start. I'm going to start talking a little bit about plants as biofactories, and also comment the production of proteins for biomedical or pharmaceutical interest. Let's start going back 40 years ago to try to understand how these techniques work actually. If we go back to the middle 60s, there is an important date, 66 from the last century, in which for the first time it was deciphered the genetic code for all the amino acids. It was very important because thanks to this, it was possible to settle the biotechnology as we know nowadays. There is another two important dates, 76 and 70, where it was discovered ligase enzymes and also restriction enzymes. As you know, these are tools very important for actual biotechnology. And also, there is another important date, which is the 72s, when the first recombinant DNA molecule was produced by Paul Berg, and also two colleagues of him, Stanley Cohen and Herbert Boyer, for the first time in 73, produced the first recombinant DNA organism. And also in the 77, it was able to decipher by sequencing, thanks to Fred Sanger, all the nucleotide sequence. Okay, so this gives rise with mainly these three scientists, Berg, Cohen, and Boyer, for the first time produce a recombinant organism, which is E. coli. Is a bacteria by combination of an antibiotic-resistant genes from another bacteria, from an Staphylococcus. Well, this is very important to understand what is a recombinant protein. So recombinant proteins, as most of you already know, is a modified or manipulated protein codified by the recombinant DNA. This recombinant DNA codifies inside a plasmid. This plasmid is including our gene of interest under a promoter, which is going to do possible the expression in another organism. When we settle of this construction inside the plasmid and inside the bacteria, and we transfer all this information in another organism, in a host expression system, this host expression system machinery start working in order to produce our protein of interest. This technique provides large amounts of any protein that could be used for research, diagnostic, or even therapeutic uses, also for industry, for cosmetic, foods, whatever interest. Okay. So the first genetically engineered human protein, it was the insulin. It was produced in the 78 using E. coli as bacteria to produce this insulin. And it was for the first time commercialized in 1802 under the name of Humulin by the company Lilly. And it was the first commercial recombinant protein that was available for therapeutic use. Okay. So since this year, since 82, more than 130 recombinant proteins-based drugs or molecules have been approved. Among them, we can find the clotting factors, some vaccines like anticoagulants, hormones like insulin that I mentioned previously, hematopoietic factors, interferons and interleukins that are able to treat a huge number of diseases like hemophilia, different kind of viruses, also to treat anemias and neutropenias and other important diseases like rheumatoid arthritis, several types of cancer, among others. So since then, many of the recombinant proteins used as pharmacological interest or as medicines are created in laboratories inside cells. Most of them does not exist in nature, but are modified in laboratory in order to perform different functions, right? Using cells as biofactories for protein expression, we can talk about different systems, including bacteria, also yeast, and eukaryotic cells. Among them, we can use insect cells, mammalian cells, or even we can use some transgenic animals, and of course, transgenic plants. Let's talk a little bit about some of them. The first one is the system of bacteria, which is the system easy to use and easy to isolate. As you know, microorganisms are easier to grow in laboratory conditions, but there is a handicap, which is that the bacteria do not perform splicing and do not glycosylate and is not able to produce the post-translational modification that are necessary for an eukaryotic cells or protein. These kind of signals, glycosylations, are like labels that the cell uses to move the proteins from one side to the other inside the cells. And there are also some markers that helps to determine the protein folding, the structure, and the function of the protein. In the case of the production of recombinant proteins from yeast, as yeast are eukaryotic organisms, they perform glycosylation, but in a different way than in humans, for example, and this can cause some problem of immune response when we use these proteins produced in yeast. However, they are easily scaling up because they grow in bioreactors and fermenters. It's not very difficult to get a huge amount of proteins. In other way, we have also eukaryotic cells coming from insects, for example. With insect cell, we have two possibilities. In one side, we can grow the cells individually in a special medium, infect them with baculoviruses, which are not harmful to humans. They don't affect human cells. Or we can grow whole insects. We can grow the complete insects, and then this give us the possibility of glycosylations, but still remains a problem because usually glycosylation in insect cells is quite different from humans. Humans carry out a very complex glycosylation, and sometimes it's necessary to work with mutant cells, mutant insect cells, to try to get the levels of human glycosylations. The other system that I would like to talk about is the mammalian cell cultures. In this case, we can use cells that excrete the protein to the media, or proteins that express the cell intracellularly, and we need to break the cells to get the protein. In this system, the cells are close to the glycosylation of humans and also the post-translational modification because are more complex. but in any case, they are not identical, depending on the species. Glycosylation, for example, of men and monkey is quite different from other mammals, so we produce a different kind of terminal sugars, and glycosylation could vary in every kind of tissue or cell type. And there is also important that this kind of cultures grow very slow, and also is increasing the risk of fungal and bacterial contamination. Also, for scaling up is very difficult because most of the mammalian cells grow by adhesion, right? The other possibility is to create transgenic organisms as biofactories for this kind of protein expressions and purification. Recombinant proteins from transgenic animals is difficult to get. It takes a long time to get ready. It's a very expensive technology. It's also very difficult to maintain because it's necessary a very big and large infrastructure, and it's not always available in most of the researcher institutions. There is also the risk of contamination from animal or human pathogens in this kind of systems. And also the law in many countries is not very permissive in its use. Also, we have to take into account that there is a problem of negative connotation that the GMOs have at the social and ethical levels and governmental restriction, right? The other possibility, talking about transgenic organisms, is the transgenic plants. The plants have some kind of advantage that I will mention. The first one is that the plant-based systems are lower cost, are quick, is unlimited scale up because we can culture a lot of plants. It's very rapid. It's usually easy to produce high yield production, and has safety because this decrease the risk of contamination from animal or human pathogens in plants. And also plants have the ability to produce novel and complex molecules that in cells, animals is not possible because it could cause a problem of toxicity and however. Well, as you know, plants have been used to make medicines for centuries. Prior to plant technology, to plant biotechnology techniques, the first drugs and pharmacological molecules were extracted from plants. Some examples very common are the isolation of the active ingredient from willow tree bark, from salic, which is the acetylsalicylic acid or aspirin that everybody knows. Another example is the opiates such as codeine and morphine that are coming from poppy cultures, and they are used nowadays in medicine. Okay, so from the beginning, in the 89, it was for the first time produced a recombinant protein in plants 30 years ago. It was possible the expression of antibody in plants. This was published in Nature, and it was the first report of a successful expression of recombinant protein in plants. Luckily, in the 90s, the first plant-derived pharmaceutical protein it was produced. It was a human serum albumin, and it was produced in tobacco and potato plants. Following in the 2006, it was created the first plant-made vaccines. This is a vaccine for Newcastle disease in poultry. It was made in tobacco plant, and it was also approved and commercialized by the company Dow AgroScience. More recently, more in 2012, another company, it was able to produce the first plant cell-based enzyme replacement therapy, expressing the thalygluciferase alpha from humans used to treat the Gaucher disease. It was approved by the FDA of the US, and it was produced in carrot cells, and is commercialized under the name of Elelyso. Okay, and more recently in 2015, there was this epidemic of Ebola, and the company Left Bio, together with MAP Biopharmaceutical, developed the CMAP, which is an experimental vaccine made from a cocktail of antibodies that is working properly to treat human patients with Ebola virus. Okay, so this was very important because the FDA granted the fast track designation. And it was produced in tobacco plants and is still used. Okay. And now, in this year, recently in February, it was for the first time announced the production of vaccines against COVID-19 by medical and GSK companies, and it was approved by Canada governments under the name of Biofactories. And it was also produced in tobacco, in Nicotiana benthamiana plants. Okay, so as you can see, the demand of recombinant proteins is increasing gradually. There are many biopharmaceuticals that are being produced in plants. Some of them are already in preclinical or clinical trials. Plants are producing antibodies, vaccines, enzymes with industrial or pharmacological interest, hormones, and also antigens for diagnostic reagents, etc. So plant molecular farming is considered one emerging technique that has grown over the past 2 decades. The major factors that makes choose technology towards potential competitors are the cost effectiveness, scalability, is quite flexible, and also is a robustness. There's a robust system to produce proteins. Okay. And also it's very important that we have to take into account the development and improvement of the transient expression system. This system that we are going to talk about a little bit significantly reduce the protein production timeline and greatly improve the protein yields in plants. We are going to talk and focus in transient gene expression in plants, which is what we do in the lab. Transient gene expression is the temporary expression of genes that are expressed only for a short period of time, and this is the base of the molecular farming. This is the base of the use as plant as biofactories, and it can be used to produce proteins at a high scale, on also in basic research to perform characterization of some proteins, localization in different tissues or cell protein subcellular localization, and also protein interaction. So it's easy and quite quick performance, and is used, as I tell you, in both in basic and applied science. There is 2 important methods to transfer the genes. There is the direct method by using gene gun or particle bombardments, and also the indirect method, which is produced using Agrobacterium tumefaciens or plant viruses. We are going to focus in this one, in the one using Agrobacterium tumefaciens. Okay, so before continue, we need to understand how we use this technology. This indirect transient delivery exploits the ability of some bacteria present naturally in nature that has the capability of transfer part of their genome to plant cells. In this case, Agrobacterium is naturally present in the soil, and it has a plasmid that contains several information, and it has a small region called tDNA, which is able to transfer to the chromosome of the host cells. If the plant suffers a wound or injury, the bacteria can infect the plants. Is able to integrate this part of the plasmid into the chromosomal of the plant, and the plant, it will start to produce some metabolites and some proteins of interest for the bacteria, for the Agrobacterium. Among these proteins, it produces factors that develop this kind of tumors that are called Crohn-Gall disease. As you can see here, this is a growing cell mass that is produced in the plants after being infected with agrobacterium. It's affecting most higher plants in one way or other. You can see here some examples: salix, tomato, and also Arabidopsis. And what the bacteria do is to transfer, as I told you, this kind of genetic tDNA, this genetic information to the host plants to make the plant produce oncogenes and genes to synthesize some kind of metabolites that the plant use to feed, to grow, and to use in their own metabolism. What we do as biotechnologists is to remove these genes from the plasmid in order to impede the plant, once it's infected, to synthesize these genes, and the plant no longer produce tumors and no longer produce this kind of metabolites. But in contrast, we can substitute this kind of genes for our gene of interest or some selection genes like antibiotic resistant genes, or some other marker like GFP or whatever marker we are interested in. And what we do is to integrate our genetic information in the plasmid, in the Agrobacterium plasmid. When we clone our gene of interest, we put the plasmid back into Agrobacterium, and with this Agrobacterium genetically manipulated, we infiltrate the plants. We transfer this information to the plant. This method is called agro-infiltration, and is the method that we use in the lab, and we are going to focus a little bit in this technology. Well, this groundbreaking technology established this method. This method for rapid recombinant protein expression is quite flexible and is scalable. Since agrobacterium strains are disarmed, as we have removed all the genes responsible of the tumor forming and metabolite synthesis. Also, another advantage is that we do not transfer the genes to reproductive cells. In this case, we don't modify the germ line. We don't perform transgenic organisms, because this is transitory expression. And this means that this technique, it could be considered environmentally safe, okay? We can use whatever vector, whatever plasmid available in molecular biology, always that has a replication origin for Agrobacterium. In this case, we have strains of Agrobacterium that already contain a plasmid with all the information necessary to transform the tDNA to the host cell, to the plants. And what we do is manipulate another vector called binary vector. We manipulate this vector in the lab, cloning our gene of interest, marker, or whatever we are interested in. And then we put it back into Agrobacterium. What we do now is to transfer by electroporation our plasmid manipulated into Agrobacterium. All of you know this technology. We select positive colonies by resistant to some antibiotic, usually. And when we find the positive clumps, we grow the culture. First, we grow pre-cultures, and after that, we grow high-scale cultures in order to get enough volume to transform the plants. We transform the plants by agro-infiltration, which is the technique I'm going to explain, using a syringe, small syringe, and pressing into the underside of the leaf. I will show you how. In this way, what we do is to transfer all the genetic information present in Agrobacterium to the host cell. For do agro-infiltration, we need to know a little bit about leaf morphology, right? If we make a sectional cut, we can observe among the two layers of epidermal cells, there is another area called mesophyll. In this place, there is an interspace that usually is full of air. It's called the apoplast. And what we do by agro-infiltration is to transfer our suspension with agrobacterium to this area, to this apoplast in the leaf, removing the air and put in contact our culture with agrobacterium with the cells of the plants. In this way, what we are doing is to favor the infections. In this process, we grow the agrobacterium in a different medium with some kind of phenol compounds and other molecules that favor this infection and this transference of tDNA in order to produce the recombinant protein by the plant. We do, as I told you, with the help of a syringe, pressing very carefully in the underside of the leaf with the syringe without a needle. And I would like to show you just very fast and a small video in where you can see how we do this technique in the lab. This is not our lab, but it's very illustrative. As you can see, we put the syringe with a needle, doing a little bit of pressure with the finger in the other side of the leaf, and we are releasing very carefully the medium containing the agrobacterium cells with our protein of interest. As you see here, it looks very easy to agro-infiltrate, but you can ask our students in the lab and you will see that this is not so easy. It requires a lot of practice and expertise to get this level of agro-infiltration. As you can see here, all the leaves have been infiltrated, right? Well, once we have done this agro-infiltration, we already have our DNA into the plant cells, and the plants start to synthesize our protein of interest. We calculate 1-2 days or even 3 after the infiltration to start collecting the plant extract. What we do is collect all the biomass or the foliar aerial biomass of the plants. We prepare plant extracts, and from this plant extract, we purify our protein of interest, our recombinant proteins, right? We in the lab are using the model species Nicotiana benthamiana, which is tobacco plants. It's of common use in biotechnology laboratories. It's very interesting because it has already the genome sequences. It's easy to maintain, grow fast. It has appropriate laboratory size and biomass. And also it has a low immune system activity. So it has a very high capacity to express transgenes transiently. As we work in the faculty at the Department of Biochemistry and Molecular Biology, we are growing our own plants here at the department. And we work in the group of allergens. So our main objective, it was to develop this system for the production of recombinant proteins in plants using this model species, Nicotiana benthamiana, in order to identify and characterize some allergens and other proteins to perform new diagnostic techniques and also personalized therapy against different kinds of allergies. We start cloning different genes, different allergens genes. We start cloning the CUP-A1 and the SALC-K1, which are peptide liase and pectin methylsterase. They are coming from Cupressus arithonica and Salsolacali, which are plants that produce a very high allergens with very problems, with a lot of problems for allergies. And we try to purify them from plants. There are proteins around 43 kilodaltons, and we are setting up the cloning right now in order to start to produce these proteins. But first of all, to setting up this system, we start producing the GFP. What we have done, it was try different constructions with the GFP. We clone into different plasmids, we try different strains of agrobacterium, and we agro-infiltrate the plants After a few days of expression, we detect the GFP presence with a UV lamp. We also harvest protein to do protein extraction and try to quantify and do kind of kinetic expression of GFP. And what we observed, it was the expression of the GFP. Here I show you some images of our genes green fluorescent plants. As you can see here, those are Nicotiana, benthamiana plants expressing the GFP at different levels. We can compare different levels of expression of this GFP. Here, there are some other examples. And what we are doing is trying different plasmid with different constructions, different sequences in order to improve the production of proteins. This last one is the one that is working better and is the one we are working with to produce the allergens. And just to let you know, by using this technique, this is a GFP expression with without histidine as tag to purify the protein. And using this technique and this kind of plasmid, the peak plasmid, is able to produce grams of recombinant proteins per kilogram of fresh weight of plant, of fresh aerial part of the plant. Okay. So this is being used to produce a lot of proteins of pharmacological interest. And we are also involved in the production of, apart from the allergens, we are trying to produce some antigens to develop vaccines against COVID-19 and detection tests or whatever proteins related with this this pandemic. Because we are part of a project, a REACT project from the European Union and the Community of Madrid. I belong to this project thanks to the SFUM-PROT group from the Faculty of Chemistry. And we are developing this kind of proteins in different systems, among them plants, of course. Well, just to finish, I would like to open the door to any kind of proposal for producing proteins using plants as recombinant proteins production system. We can try, try to produce in plants. Having said this, I just want to thank you for listening me, and also would like to thank especially to my undergraduate student Lorenzo Navarro Sanchez, who was helping me in the developing of this technique here in the lab. It has been hard to get ready everything because we are working in a department where nobody works with plants. And it's taking time, but we are getting very good results. So I want to thank him publicly. And that's all. Thank you, all the audience. I'm happy to answer any question you can make. Thank you very much.
Thank you, Mercedes. Great presentation and very interesting. I don't know if the audience have any question.
I have several. Mercedes, I think that was a nice presentation and a very interesting work. And I think that it's also very promising work with many applications. I want to ask you about the nature of the proteins that you can express in this kind of system. For instance, which is the low limit of you can express, for instance, peptides or small proteins, or do you have a limit in the size of the molecules that you express in this system?
Okay, well, there is not a problem of size. We can try with every kind of proteins, from small peptides to complex proteins. There is usually not a problem of size. It's depending on the kind of plasmid that we use. If we are trying to produce a big protein, we need to use different kind of plasmid that favor this big proteins folding. If we are talking about a small peptide, it's going to be easy, of course, but we could try with whatever kind of protein, whatever size. Of course, if it's a very complex protein, it's going to take longer, and we will need to improve the techniques probably. But at the beginning, it's possible to try whatever protein we are interested in.
Okay.
So this is not a problem of size.
Then I think that is something that we have to know, that if it's any kind of protein, any size.
Yeah.
I have another question. I mean, sometimes one of the problems when you express the protein in a bacterial or a yeast expression system is the toxicity of this protein. Sometimes the protein is toxic for the cells in which you are expressing the protein, no? How, I mean, the plants have this problem, or how you can avoid the toxicity of a protein in order to express the protein in plants?
Yeah, this is a very important question. Usually plants present an advantage to produce this kind of toxin proteins, because sometimes there are proteins that are toxic for mammalian cells or animals, like if we are talking about pharmacological proteins. And these kind of proteins usually are innocuous to plants and is able to produce them in plants without any problem of toxicity for the plants. In case that there is a toxic molecule that could affect the plants, we have to take into account that we are talking about transient expression. It's going to be expressed for a short period of time. So if the protein is toxic for the plants, it's able to produce it and recover it before the plant start to be damaged. So it's working properly with some kind of proteins, for example, insecticide or venoms, or it has been producing in plants without problem of toxicity, since we don't allow the plant to develop after the infiltration. Okay. Anyway, if there is some kind of toxicity for the cells, we can use some kind of promoters to develop to direct the protein to a organelle. For example, the chloroplast. It's possible to direct the protein to chloroplast excretion. In this sense, the protein is going to be contained into the chloroplast, and we can purify the organelle and after extract the protein from the organelle. This is also possible.
Okay. Well, I have another question in terms of the amount of protein. You say that you used, I mean, you estimated grams per kilograms of the wet material, no?
Yes, fresh weight.
Okay. Then which kind of, I mean, if you want to express, for instance, 1 gram of a protein, how many plants of these that you have in the lab, how many plants you can grow in order to get 1 gram of a protein, more or less?
Yeah, well, it's difficult to say because it's depending on the protein. There are proteins that express very well, and we can get a huge amount of protein in the order of grams per kilograms, as I told you. There are others that are more difficult to express, and we get less quantities. But in any case, it's easy to scaling up. If the protein is difficult to get, we can grow more plants and purify from more biomass, fresh biomass of the plant. I mean, we can transform 5 plants, or we can transform 50 plants. And the problem is the purification. If we are producing a protein with a tag, for example, a his tag, and we can concentrate this protein, it doesn't matter if it's lower expression level, we can infiltrate more plants and get higher quantities of proteins. There are also, I didn't mention because it's more complicated, but there are some sequences included into the plasmid that increase the protein levels. We are working in the lab, especially with the HC Pro sequence. I didn't talk about this, but it's very interesting because it's a sequence coming from viruses, which make possible to silence all the immune response of the plants. The plants, when are infected with some external DNA, develop a response, immune response, kind of immune response, and the plants start to silence this exogenous DNA. And there are sequences coming from viruses that are able to inhibit this silencing process. And in the case of difficult proteins, we use this plasmid with this sequence, which increase greatly the amount of protein that we can get. We are working in the lab with one of these plasmid, and we have checked and we have seen differences in the presence, for example, of GFP by using this sequence or not inside the plasmid. And we are getting promising results, and we're expecting to use it also with the allergens and the COVID proteins.
So, I mean, I think that it's a wonderful work, and I think that for allergens that are, I mean, from vegetables or from plants, I think that it's going to, I suppose that their yield is going to be much better than in other systems. Then I think that it's a very good. I mean, I think that your introduction in our group, it's wonderful. I think it's wonderful for our work, for our to enlarge the number of systems that we have to express proteins. And then I think that.
Thank you.
Nice.
I want to take the opportunity to thank all the group of all the allergens group, because well, you are helping me a lot at the beginnings, and I'm very happy to be
but
here.
There has been a symbiotic relationships, and I think that it's nice. The only thing, I have only one curiosity in terms of which is the difference in terms of yield, or which kind of proteins when you use the gene gun or you use the agro-infiltration. Which is the difference with both systems? So, there is one that is better for one thing, or no? I mean,
yeah, there is a very big difference. In the case of the gun system, the particle bombardments is a let's say root technique, because what we do is directly to shoot the DNA. Is the DNA coated with a metal particle, used to be gold or tungsten or another metal, and this is used as a carrier with the plasmid, with the DNA, and direct shoot into the cells. This could damage the cell, and the amounts of production of protein is very low. This system is not usually accepted for recombinant protein productions. You can use it to do transient expression, just to, for example, to follow a protein at the localization level, so cellular localization with a GFP or another marker, or to see interaction protein-protein or protein another molecule, but not for recombinant production of proteins, because it's going to produce a very small quantity of protein. So it's not valid for this method.
Okay, thank you very much. I don't know if Carlos has more questions.
Yes, I have a question, but I see there's no question by the audience. Mercedes, why permanent transient or expression and not permanent expression?
Well, it's transient expression because when we do the agro-infiltration, we do not transform any germ cell. We only transform somatic cells, because we are introducing our bacteria into the leaves, and these are somatic cells which are no longer to transfer the genetic information to the descendancy. So we are not performing a stable transgenic organism. It's just transitory. And after the agro-infiltration, we collect all the biomass, all the aerial part of the plant, and with this material, we perform the protein extracts. So we destroy everything. This is never going to be transferred to the descendancy. So it's environmentally safe, because the transgenes never pass through the descendancy.
But are there stable methods, methods to a stable expression in plants or not?
Yeah. There are, of course, we can generate transgenic plants for recombinant expression. But depending what you want to do, if you need to produce a high-scale protein, it's better to do transients because you express the protein, take the material, purify the protein, and that's all. If you need to generate a transgenic, for example, with any kind of modification like insect resistance or herbicides tolerance, or even include a trace for nutrition properties or whatever, you express a recombinant protein constitutively along all the life of the organism, along all the biological cycle of the plants. So this is a stable transgenic, is an OMG plants, but this usually for a different purpose. If you want to get a high-scale protein recombinant production, it's better to use transient expression systems.
Okay, and another question. In the chat, there are a question, no? Saracen Bain Santos, how to promote sustainable chemistry with transition methods?
No.
Ah.
No.
¿Esto qué es?
Eso es la charla del próximo día.
Ah, vale, ok. Another question, Mercedes. You answered a question about the immune system in plant, but what that recombinant proteins are able to perform an immune response. What are these mechanisms or the immune response in plant?
Well, this is an interesting question. When the plants perceive some kind of external factor, that could be an infection, or in the case of agrobacterium, is a transfer of a tDNA, is an exogenous genetic material. Like in the case of plant viruses, as you know, the virus infect the plants, trying to transfer genetic information to the plants. And the plants develop a kind of immune response. It's not exactly an immune response like in mammals, but trigger a kind of mechanisms to try to avoid the presence of this external factor. One of the first that they start producing is the silencing of genes, the silencing of the messengers. As the usual method with this kind of dicer proteins to try to degrade the messengers and cut in a different pieces in order to try to block this enter of exogenous DNA material. There are viruses that are able to avoid this silencing mechanism developed by the plant, and this is a very good strategy that we use. We are able to clone these virus sequences into the plasmid that we use to produce proteins in order to silence this response of silencing coming from the plants, and this allows us to produce a higher level of protein in this system.
But silences is the unique mechanism, or there are more mechanisms?
There are more mechanisms, and there are different ways of silence these genes.
Oh.
It could be by silence small RNAs, microRNAs. There are many different systems, like in mammals, but more specific of plants, but basically it works in the same way that
okay.
Animals.
For me, no more question. No, I don't know if one question.
It's not a question, it's a admire. Yeah, because it's something that say that it, from the University Sorbonne, I mean, from the Sorbonne University. Somebody that it's said that it has been an interesting work and showing in a very fascinating way. Then it's one admire that you have, Mercedes.
Thank you very much. I'm happy to hear that.
Well, I thank everybody for attending this seminar. And I have to announce that the next one is going to be May 19. Sarah Zembeji from the Organic Chemistry Department is going to talk about how to perform sustainable chemistry with transition metals. And it's a very interesting topic too. Then see you to this day, to everybody, and thank you again to both of you.